Hello, can everybody hear me? I see Konrad, I see Karim, this is awesome. Okay, awesome, everyone can hear me. I see a lot of yes. So yes, I see that you have already figured out how to put your comments in the chat box. This is awesome. And then I just also want to quickly mention that on your top left corner, you can see different sessions, and this is for if you want to go to a different session. And after each session, the recording will be posted in the link where they are live streamed. And then at the bottom, you can see ask a question, and you can see post. We don't really have any posts yet, but you can definitely post a question. And you can also upvote questions that you are also curious about. And that's just going to allow the speakers to see the most upvoted question at the top so they can answer that. Okay, so let's get started. Welcome everyone to USS2020, which is the Unique Student Symposium. And I just want to start by introducing UNIQUE, which stands for Unifying Neuroscience and Artificial Intelligence in Quebec. Let me just quickly focus on my slides. Okay, and so UNIQUE stands for Unifying Neuroscience and Artificial Intelligence in Quebec, and we're just a research center at the crossroads between AI and neuroscience research funded by FRQ&T, and we have over 80 regular and collaborating researchers from leading academic institutes inside and outside of Quebec, spread over 4 research axes. There's perception and control, higher order cognition, learning, and neural networks. A key role of UNIQUE is to promote bidirectional research between scientists in neuroscience and AI, and to establish a high-impact neuro-AI research discipline in collaboration with local and international partners. And our vision is to develop a one-of-a-kind interdisciplinary research and training strategy at the crossroads between AI and neuroscience. And of course, I would like to take a moment to thank my amazing colleagues who organized such an amazing event as Stephanie, Bertha, Pravish, Surya, and Naz. And of course, the Unique Student Fair Committee, Jess, Annalise, Raymond, and Elizabeth, who gave us a lot of support during this whole organizing process. And of course, on behalf of the organizing team and the Student Fair Committee, I just want to say thank you for all of you who showed up at like 9:30 a.m. on Saturday morning, and for showing us your passion about neuroscience and AI. And personally, I had the most fun organizing this event with my colleagues, and we're just hoping to present to you a really fun day where you can learn some neuroscience and where you can learn some AI or a bit of both. And we hope that you enjoy this event as much as we did when we're organizing it. And as some of you may know, this event was originally planned as an in-person symposium in Montreal. But given the current circumstances, we had to shift it online. But of course, the silver lining of that is it's more accessible for those of you who are outside of Montreal and Quebec. And of course, there's the benefit for climate change and environment. Regardless, I still hope that all of you and your family are staying safe and healthy. And I hope that this symposium can give you an opportunity to stay connected with the science community in this self-isolating time. So we have scheduled a social event / coffee break right after our first talk, and we'll be using a platform called GatherTown. This is just something that they used during iClear, and it turned out pretty well, and we thought we would give it a try. So hopefully that's gonna be fun. Okay, our agenda. So we have 2 keynote speakers in the morning, Dr. Kording and Dr. Muller. And in between their talks, like I mentioned, there's this coffee break / social event. And in the afternoon, we have another keynote speaker, Dr. Fairhall, and she's going to give us a talk on biological dynamics and their role in computation and learning. And after that, we're going to take a little break, and we're going to have our breakout sessions that are arranged according to different topics. And you can find all the instructions in the emails I sent to you before this event started. And I also just want to kindly ask you, throughout this day, please chat in a professional and respectful manner. Please refrain from harassing people or using crude or inappropriate language. Okay? And, well, hello from Berlin. Nice. Okay, So our first speaker is Dr. Konrad Kording from University of Pennsylvania to give us a talk on whether the brain does something like back propagation. So Dr. Kording obtained his PhD in physics at ETH Zurich in 2001, and he then worked as a postdoc at ETH Zurich and UCL, followed by a Heisenberg Fellowship at MIT. He was a professor in physical medicine and rehabilitation, physiology, and applied mathematics at Northwestern University before he joined the faculty at the University of Pennsylvania with joint appointments in the Department of Neuroscience and the Department of Bioengineering. His research focuses on the application of emerging data science techniques such as deep learning for the analysis of neuroscience data. More recently, he is interested in causality and its links to machine learning, as well as understanding the credit assignment process that the brain does. So I'm going to invite Dr. Kording on the screen. Okay, hopefully, yes, he is accepted. Awesome. Hello.
Hey, good to see you.
Good to see you. You sound awesome.
Right. Great.
So, do you know how to share screen? Can you do that?
I most certainly know how to share my screen. Give me a second here.
Awesome.
It's as you may know, I ran this other thing called Neuromatch, and I had to grapple a lot with the same kind of problems that you guys now have to grapple with. Okay. So everyone, you need to appreciate how much work goes into this. And before starting my talk, I first want to thank the organizers. This is incredible what they're doing. And I think it's so important for us to basically stay connected, stay a community at the times of COVID-19. In fact, I personally was looking forward to spending much of the summer in Montreal this year, and it's not going to happen. So I'm happy that we'll be able to stay connected like this. So how does my screen look like? Is it properly shared?
Perfect.
Great. In this case, should I take it away, or what's the plan?
Absolutely, please. Everyone, welcome Dr. Konrad Kording.
Great. Well, thanks for you all to listen in. So my talk today is entitled, "Does the brain do something like backpropagation?" And I want to ask one thing of the organizers. If there's any questions arising or something, I will not be able, the way I screen share here, to also monitor the chat. So in the questions. So if there's a question in between that is really important at that point of time, please feel free to interrupt me at any point of time. Okay, great. So let me jump right back in. So let me jump back in with the negative view that I hear from some people. You know, like people say, well, a long time ago, people were thinking of brains like hydraulic things, where they said, well, hydraulic is the best technique that we have, and we can do all kinds of things with hydraulics. Let's think of the brain as like a hydraulic device. And then later on, they started building simple electrical circuits. And then they were talking about electrical brains. And then computers got popular, and the computer metaphor took off. And now deep learning is popular, and people say that the brain might be like deep learning. And that's the cynical view, where it's basically, it's just an opportunist thing. Deep learning is popular, so we're just basically like surfing that popularity. Over the talk today, I hope that I'll manage to convince you why I'm not taking that cynical view, why I actually believe that deep learning has a lot to contribute to the way that we study neuroscience. So the question of today's talk is, could the brain do something like backpropagation of error? And before I go into why the brain could, let me just zoom out all the way. What does it mean to have a learning algorithm? Well, for me, having a learning algorithm just means that we have a system that has parameters. Let's call them W, and the system changes the parameters. So I experience something new, and in response to that experience, I change the parameters that I have. I have a delta W. Anything that does that is a learning algorithm in my view. Now, let's talk about examples of learning algorithms. Now, on the machine learning side, backpropagation and all its variants is one set of algorithms that we use, or reinforcement learning. Then on the more biological side, we have Hebbian learning, which is an abstraction of some of the physiological data. And more on the biological side, we have neuromodulator-enabled Hebbian learning, which means that basically the change in the synapse depends on what the presynaptic neuron does and what the postsynaptic neuron does, and basically a global signal that says, hey, this was awesome, or that was pretty bad, you don't want to do that again. And it also includes biological things that are far more on the biological side, like homeostatic plasticity, which is this phenomenon that's been extensively studied by a number of people, including, above all, Gina Turriciano, which says basically that if a neuron hasn't been active for a long time, it tends to upregulate its synapses so that it will be more active, and if it's very active, it tends to downregulate them, called homeostatic plasticity. But the important thing is, in this view, basically biology and technical algorithms are like the same thing. They're just changes in some parameters. So let's see what we can generically say about learning algorithms. So there's one general observation, which is good learning systems improve. What does that mean? We generally don't get worse at things. Yes, there are cases, and we study them extensively, like Prism adaptation, for example, where you learn something and you get worse at something else. But by and large, whatever we learn, we kind of get better in life, including we learn something and we, in fact, get better at other things. Whereas a lot of technical systems, actually, they learn a new thing and they get worse at other things. Most importantly, learning systems, good learning systems, including essentially all biological systems, generally get better at the things that they are learning. No? And machine learning systems also, of course, get better at the things that they're learning. So under which circumstances do learning algorithms work? I will now focus on learning algorithms that learn by making small changes. And mathematically, what I'll be talking about for like 2-3 slides, this is an absolute truism, but it shows a certain principle that is shared by all those systems. So if we have a loss, which is a measure of performance, then the change in loss, as long as we talk about small changes, the change vector in parameters, delta W, multiplied with the gradients of the gradient of the W of the loss. Okay, so this is generally the case. And now let's briefly review why backpropagation works. Now, backpropagation says that the weight that we induce is minus eta, a small constant, times the gradient of the loss of the weights. The improvement then is the weight change times the gradient of the W's of L. And now we just put in the equation from backpropagation, and we'll see that basically we have minus eta times the gradient of the loss function transpose times the gradient of the loss function, which is always going to be positive. No? And that's why backpropagation generally leads to a decrease in loss. Okay, that's why backpropagation works. Now, the question here, and I think there's a broader principle. No? Like, there must be a reason why also biological systems get better. And this equation about the improvement, that the improvement is basically the scalar product of the gradient with the weight change, is true just as well for biological systems. So somehow if a biological system gets better over time, just like gradient descent, the changes that it does must correlate to the gradient of the loss function. Okay, now the field has tried all kinds of non-backpropagation ways of optimizing things, and a lot of those don't usually work. So here's for example, a simple idea. So we have a system, it has lots of synapses, and it's known in biology that synapses are stochastic. So if you have an input coming into a synapse, sometimes it produces an output, and sometimes it doesn't. It's what's called failure. We have a release when actually this stuff works, and a failure when, for some reason that we're still trying to understand exactly, it doesn't work. And now the idea is, well, what if we take the times where a synapse was off, it was a failure, and we see how good the outcome is for the animal. And we take the times where it was a success, and we see how good the outcome is. In principle, we can calculate the gradient correctly by just comparing these two situations, where I can say, well, would it be better if I wasn't there? Well, if I'm not active and behavior generally leads to more reward than in cases where I'm active. So therefore, it would be good if I turn myself off. And that idea can be implemented, and it can be proven that it does the right thing, that it will eventually optimize things. So what's the problem with it? No, and like people thought about what the mechanism could be. We would need an eligibility trace. We would need this random component, and we'd need a reinforcement learning signal. All the biological components are readily there. No, we have neuromodulators that tell us how good things are. We can relatively easily imagine that in a synapse, we could have a calcium signal or something that tells us if the last one was a failure or release. So the components are there, but what's the problem there? So we can ask, what's the variance of that way of learning? So let's say we take all the synapses in the system and we introduce some noise to them. Now, like we basically, every trial, every short period of time, maybe 10 milliseconds, maybe 100 milliseconds, all the synapses are basically randomly perturbing themselves, and we can model that as a Gaussian. The weight change is a Gaussian with like epsilon variance. And then we can say, well, let's as long as we have small perturbations, we can locally Taylor expand that, and so we have therefore that we have the weight change, the randomly induced one. Now delta W is a randomly induced one, not the learning one, times the gradient after I, and now so this is finite size differentiation, effectively differentiation from small differences. Now, but we can also calculate what is the variance of the estimator that we get. Now, every synapse that we perturb like that induces variance on the output of the neural network, and therefore the variance of this estimator goes up linearly with the number of weights that we have there. In other words, for any big system, this is absolutely not usable. It would be incredibly slow to learn like this. And we know that biology is pretty fast at learning. So based on this idea that we can't seem to find ways of allowing fast learning, efficient learning, without assuming things like backpropagation, there's the assumption that there may very well be an algorithm that says that the way the brain changes over time is like the gradient of some cost. And now the question is, could the brain implement something like the backpropagation of error algorithm? And there's two problems that people talk about. The first one is the weight transport problem, and the second one is the multiplexing problem. In short, for things like backpropagation, we need to propagate the signal forward and we need to propagate the signal backward, but we need to use the same weights in both directions. The second one is, if we have something like backpropagation, we have two different kinds of signals that mean something very different. We have the activity, and then we have gradients, and they are very different. And I should first mention that the quest for biologically realistic backpropagation is about as old as backpropagation itself. So here we have a paper from 1985, and there's a lot more from that era, a learning algorithm for Boltzmann machine, which was, again, meant to give biologically realistic ways how the brain could be implementing something like that. But let's first talk about the weight transport problem. So we have an input. We have a hidden layer. We have a W0 that goes from input to the hidden layer. We have W that goes to the top. And if we run backpropagation, we have W transpose on the way back. So here is a rather shocking paper by Tim Lilly-Krepp from the Tweet group back then. He moved on to be at DeepMind then. So what if instead of doing good assignment, basically using the right weight on the way back, which we need for backpropagation, what if we just ignore the problem? No, like what if we just say, let's use a random matrix B? Maybe we don't have a way of producing weight symmetry. Maybe let's just ignore weight transfer. And people knew that that was absolutely impossible, but for some reason the Pram doesn't know that this is impossible. So what they have here is they show that if you do that, in a lot of cases, you're about as good as if you do backpropagation. For example, if you look on the left-hand side, you see learning curves as a function of the number of examples, and you see feedback alignment, and it goes down very quickly, very comparably to backpropagation. Why is this happening? It's happening because of what's called feedback alignment. So basically, the neural activity starts correlating with the gradients. It starts to be that through the learning algorithm, the forward weights make it that the backwards weights are approximately right. And that's what you see on the right-hand side. The angle between those two relevant vectors is going down over time. And then people applied that to realistic examples. So here, for example, we have MNIST, classical toy example that we use to test our algorithm, and you can see that feedback alignment works remarkably well. Importantly, it even works in cases where traditional backpropagation is impossible. So they took a spiking system here. In such a spiking system, it should no, there's no gradients. It's all spiking. It has all the things that really frustrate us when we do computational research. But so basically, we have a backward propagation credit assignment that is really wrong for what is happening on the forward side. But it works really well because the forward component learns to effectively be right, at least in first-order approximation. Okay, so then here I will briefly talk about the paper of Ben Lansdale, postdoc in my lab. So what if having not from Lillicrap's work, we know that it doesn't matter that we're really good at the feedback weights. Now the next question was basically, well, could we do even better if we learn it? And now the idea is that we do this algorithm that I told you is absolutely hopeless, the one where we locally perturb activities, because the learning the feedback weights is actually relatively easy if you think about it. Because in general, you can say, my brain, I need to learn all the 10 to the 15 or so parameters of that. Hopeless if we use perturbations. But if we just look locally at a neuron and its postsynaptic neurons, that's like a small number. That's like maybe a few thousand of them. And from Lillicrap's work, we know that if we're slightly wrong, it doesn't overly matter. So Ben basically implemented such a perturbation strategy. And again, he finds that it helps on MNIST. So here you can see feedback alignment in gray, and then you see various versions of so-called the node perturbation algorithm. It does a little bit better. And a place where it really makes a big difference is MNIST autoencoders. And where we have Adam is what you see on the left-hand side, and backpropagation. And you can see that node perturbation at the bottom right does much better at producing meaningful images of digits to feedback alignment. And in this case, we can also quantify what that loss there is. And what you see here, and I apologize, there's too many items on it, but we basically, in solid, we have test set performance. In dashed, we have training set performance. And we can see feedback alignment. It does rather poorly here. Hey, we obviously chose this task because it's one where feedback alignment isn't sufficient. Now, feedback alignment seems to work well for a whole host of problems. But of course, if we want to introduce a new algorithm, we better show that it's better than feedback alignment. So we had to push towards somewhat more complicated problems. And here, the autoencoder is one example of that. And now, I want to ask something of the organizers that's a little embarrassing, because I didn't bring my phone here because I was worried it would ring, and because I'm full screen presenting, I can't see the clock. How much more time do I have for my talk? Okay, I'm not sure. I hope that you guys still can hear me, but I can't hear you, which is a little worrying. Okay, let me briefly check that. Okay, fantastic. So I do have plenty of time. I apologize for that. We're still getting tuned to this way of presenting, and I'm making so many mistakes in different settings. In any case, so but what this shows is that if we compare even with Adam, then node perturbation does surprisingly well. In particular, Adam does relatively poorly on the test set. And it looks like node perturbation does relatively well here. I should also say that node perturbation idea here, which is really just a variant of the feedback alignment idea that Lillicrap introduced, works reasonably well for reasonably non-trivial problems like CIFAR10 and CIFAR100. I should also say we now have mathematical proofs that show that there's situations in which node perturbation converges to the right values. It's not overly surprising if you think about it, but it's nice to have that safety. So here I briefly want to plug a paper by Blake Richards' group, who is in Montreal, and therefore, like, I thought you guys should all know about it. So the idea then is, can we, instead of using perturbations, and perturbations are bad, to remind you, because introducing perturbations means that we introduce noise, variance, on the output. And if we introduce variance on the output, that makes us worse at solving our task, and also worse at solving the estimation problems. So in this very clever paper by Jordan Gregoff, he replaced basically the node perturbation things that Ben was doing with something that exploits the unavoidable noise that is induced by a spiking mechanism, and that in a way even the noise can go away there. The important thing here is that, again, like now without perturbations, they are able to rather considerably improve performance on, say, CIFAR10 and other settings as well. Beautiful work. I highly recommend you check it out. So let's summarize weight transport. In my view, weight transport may not be such a big problem after all. It might be relatively easy to learn. And so the first thing is, it's not really such a problem. Like, if the feedback weights are slightly wrong, it doesn't matter much. And the second one is, well, even if they need to be right for certain tasks, it's relatively easy to learn. Now that means that, like, one of the traditional criticisms of deep learning as a model of how the brain actually works just more or less went away. It's easy to learn, and it's not so much of a problem in the first place, which gets us to the second problem. So the problem of multiplexing. So spikes are supposed to transmit the signals, but then how could the same spikes also transmit the gradients? And like, can I know that this spike just tells me something about the gradient, or that this spike tells me something about what I'm trying to calculate? And in a way, like to put it into a nutshell, people say, like, no one has observed gradients being transmitted. So what are the ideas? So the central idea here is we somehow need two different signals, and we need to know which is which. And there are ideas that are tied to specific aspects of a presentation. For example, there's the idea that the spikes, the regular spikes, when neurons produce single spikes, that tells us about the signals that we're processing with. But bursts, when neurons in a very short period of time produce a bunch of spikes, tell us about gradients. There are other theories that say, well, if a stimulus comes on, and maybe instead of coming on, the brain is oscillatory and like it switches between them, then in that one period, we do calculations of what's going on. We do processing, and then there's another period where we transmit gradients. An extreme version of that would be, say, the wake-sleep algorithm of Hinton's, where you can say there's one very long period of time where we like look at the statistics of the world, and then another very long period of time where we instead look at the where we sort of solve the other part of the learning problem. So then there's more physiological proposals. Now, it could be that the difference between excitatory and inhibitory neurons tells us about gradients, and the activity of excitatory neurons tells us about what we are calculating. And then there's a radical idea. Well, what if there is no signal that says if it is about processing or if it's about gradients? What if it's rather that everything is learning and somehow the neurons are learning to learn? Just to give you an example of such a flavor, here's a very nice paper by Sen and Benjo, where you can say neurons have two sites of synaptic integration, apical dendrite on the upper part of that pyramidal neuron there, processing at the lower part, and we have inhibitory neurons, and we have top-down connections onto the inhibitory neurons. If you set it up the right way, it provably does the same thing as backpropagation. And in this space, there's a whole bunch of proposals there. What's going on here? Ugh. Okay, I just briefly want to mention something there, which is the spikes versus burst transmitting thing. I have a lot of love for this kind of model because that's what I did during my PhD thesis, where based on the work of Andrew Larkham, we tried to talk about how individual neurons could use spikes versus bursts to decide if they should learn from it or if they should use it for processing. Now, I briefly want to mention ideas of learning to learn or learning how to learn, because I think it's a different way of thinking about the same ideas in a way that I think is very important. Let me first highlight that people actually do such things. So this is old human psychophysics work that we did. It's a very weird task. So what you do is you draw a trajectory with your hand, and then the computer tells you how many cents it's worth. And you're trying to make as much money as possible because neuroeconomics people like if money is involved, okay? So what you do is you get basically paid by the highest score that you get, and you get like 25 trials where you can over time improve. Now, in the way we are paying people, there's two things that matter. The one thing is which direction are you moving on average? And then the other one is, are you curved rightwards or are you curved leftwards? And over time, people get really good at solving these kinds of problems. Importantly, though, we have two situations. We have one case where the angle matters an awful lot and the curvature is relatively irrelevant, and another case where the curvature matters an awful lot and the angle doesn't matter so much. And it turns out that people massively change their learning strategy between these two situations. If it's in the alpha situation, the angle matters, they very quickly get good at alpha, and if the curvature matters, they get very good at the other one. So people readily do this kind of meta learning of learning to learn. And I should mention here, this is a nice paper by Birch et al., where what they did is they put you into a world where it's good to learn quickly, or they put you into a world where it's good to learn slowly. And if you're in the world where you should learn quickly, you learn quickly. And if you're in the world where you should learn slowly, you learn slowly. So there's clearly an aspect of optimization that is learned. And in that sense, you could say, what if in each neuron we have a teacher and we have a student, where you could say the student does everything, but every neuron, and that could be the apical dendrite in cortex, or that could be the climbing fibers in cerebellum, every neuron kind of produces its own learning signal, and it basically learns that using maybe perturbation, using reinforcement learning type things, or whatever algorithms are good for that. In that case, there is no specific substrate for gradients. It's just that we have a whole part of the system that learns to properly deal with gradients. Now, this brings me to my takeaway slide already. So the argument for backpropagation, the background argument, is primarily at the moment that we don't know other algorithms that scale well. There may be classes of algorithms that scale extremely well. We just don't know that. And the concept of needing to scale well is entirely unaccepted in neuroscience at the moment. In a way, models, for me to accept a model as a candidate of how the brain works, it must have the property that it scales well, so that basically it produces changes that in good approximation fix the problem. And the arguments in the past against backpropagation in brains have primarily been of the weight transport. Oh, there's two weights, and how would that even work? Or multiplexing. How could we be doing spiking, and how could we be doing gradients at the same time? But I think neither argument is very strong. And I think learning to learn is underappreciated in machine learning and neuroscience at the moment. And in general, I think that the time is pretty good to think more deeply about linking neuroscience and deep learning. And I think we need to change. So in the past, and in this talk still, the way I talk about it is about possibilities. I told you that the brain could implement backpropagation, could approximate backpropagation. But in the future, I think we will need to shift and basically say, let's look very precisely at the brain and find out concrete mechanisms how the brain actually does it, which means not take some phenomenon from biology and kind of wave over it and say, now this approximates backpropagation if we do the math. No, like really bring it to the biological measurements. And I think this is going to be very exciting. And with this, thank you so much for listening. Now I need to stop screen sharing. How do I scrub? Here, close video, no. So we do have time to discuss, and thanks everyone for tuning in. I kept my talk short because my experience with online conversations is that they are much better than the usual offline discussions, because you can ask questions and importantly, you can vote on them. So please make sure that you now ask the questions, now in your disagreements. And you ask me those questions, and then we talk about them. And I really like that this can be very interactive. So why don't I start looking at the questions here? Yukti asked the question, how would you explain these bursts as a cellular mechanism in the neuron? Essentially, how is the gradient explained as the physiological level? Possible theories, models. This is a great question, and I'm so sorry that I didn't go through into more details with that. So in the apical dendrite, okay, let's talk about pyramidal neurons. So pyramidal neurons have a cell body. They have somatic dendrites. That are basically little pieces of wire that connect to the cell body. And then they have an apical dendrite. And the apical dendrite is this big thing going up all the way to the surface of cortex. This apical dendrite has a curious property, which is if it is depolarized beyond some level, so if there's enough current going into that, it lets in more calcium. If it lets in more calcium, the voltage goes up even more. So what happens is what's called a dendritic spike, or a calcium spike. So what happens is, just like in the axon, you have a self-reinforcing process that makes the spikes. In the apical dendrite, you have a self-reinforcing process that makes calcium spikes. So a calcium spike means all of a sudden lots of calcium comes in, which calcium by itself is a very strong gator for learning. And then the calcium signal produces, it's like a battery that is then on for like, 50 milliseconds or something. It produces a very strong current that goes to the soma. Now this current then leads to several spikes, maybe 3, maybe 5. And this set of spikes then becomes an identifiable event for downstream neurons. So there is a clear physiological mechanism how spikes are made. And now the question is, well, could this be a substrate for cellular plasticity? Now, what's going on here is that because this is a very strong ongoing current, it's relatively easy to see this current if you're a synapse anywhere in the cell, no? Because like you have this strong calcium current and you have this strong voltage that is sustained, that is out of the range of what you maybe normally have during regular spiking. And this mechanism then provides a substrate with which synapses in the basal dendrite could reach this teacher signal that is necessary for them. So this is a great question. Thanks so much for asking. So this is in cortex. Now let me tell you about another case. So for neurons in the cerebellum, they have the so-called climbing fibers, which is not two parts of the same cell, but it is another cell, in fact, in a different brain area that goes to the cell and very strongly produces a depolarizing signal. So that means that basically every synapse in that cell can again meaningfully see that reflection of that putative teaching signal. So in that sense, I think the physiology that would be needed is well established. Now, let me directly point out where the problems are. We don't know the causality of that. No one has yet been able to take neurons in cortex and vary the number of calcium spikes they have while keeping everything else the same and making sure that therefore, basically, if you block calcium spikes, there's no learning. If you make that there's far more, you have more learning. Basically, this set of biological experiments haven't been done yet, and they're very hard, but I think it would be very exciting to do that. So thanks so much, Yukti, for your question. Okay, a question from Sana. What is a Prism algorithm? So I briefly mentioned Prism adaptation. So this is not an algorithm. This is something that people use. I basically give you glasses, and the glasses rotate the entire world by 30 degrees. They are literally prisms that you have in front of your eyes like this, which means that if you move straight, it will now look as if you're moving maybe 20 degrees to the left. And people very quickly learn to deal with those situations. Cool. Okay, great. So let's see. We have a question by Joshua Benjo. Isn't the argument against perturbation also killing node perturbation, albeit with a gain of 1,000? Yes. So what Joshua refers to is there's now two versions of this algorithm that I said, I don't believe that it's a good algorithm because it doesn't scale well. In that calculation that I showed you, it's basically we have noise that is proportional to the number of synapses. There is an alternative which is called node perturbation, where instead of randomly perturbing each synapse, I randomly perturb each neuron, which is called node perturbation. And yes, this is entirely unrealistic if we talk about learning in the overall system. But if it is just to set up weight symmetry, node perturbation isn't overly a problem, because all I'm effectively doing is I spike, at least in the algorithm of Jordan in Blake's group, I spike and I basically look for the reflection of my own spike, for which not the whole brain is relevant, but basically the signals just need to get to the neurons and need to get back. Oh, actually, Joshua, you're on screen. I hadn't seen that because you were hidden by the ask a question thing. Hey, Joshua.
Hey.
Was that a meaningful answer to your question?
Yes, very good. I agree.
Cool. Great. Should we take more questions? Do you have another question? Hold on, there's lots of questions. There's 8 questions here. Would you recommend any book or MOOC for someone new to the neuroscience side of things? Actually, I have exactly what you guys are looking for. So this summer, we are running something that's called Neuromatch Academy. Neuromatch Academy is basically aimed to teach computational techniques to neuroscientists, and at the same time teach neuroscience to computational people. It's gonna happen in July. It's gonna be the biggest summer school in neuroscience ever, and it is with a spectacular group of people who are basically producing really good material in that area. Now, if you come from a computational background, this is less directly relevant for you. So basically, what you will take away isn't how to apply PCA. You know what PCA is, but it basically teaches you what it means to the biologist if you apply PCA. So you could say, if you look at the syllabus, yeah, you know the elements in the syllabus, no? You know some deep learning and you know all those things. But the important thing at Neuromatch Academy in the summer is that it really gives you the chance to kind of see the biological interpretation. So let's see, there's lots of questions here. That was a question by, okay, do you have an idea about possible physiological experiments that could support the hypothesis of backpropagation in the brain? Yes. And both Joshua and me, we have lots of ideas. So, but let me bracket that into kinds of questions. So there is one question where the question is, does the brain do something like backpropagation? What would we need to show for that? We would need to have a neuron where we know, well, the task would be better if this neuron was more active. And then we should see that the neuron becomes more active through learning. The other, so that would allow us to establish that the brain does something like backpropagation. And in a way, we know those things. And in a way, it's trivial. Like if you have the muscle neuron in my finger, and I should press the button. Well, yeah, the muscle neuron gets more active. So the question is kind of how far we can push that. But that kind of experiments establish do cells, or which cells in the brain actually go down the gradient of the behavior or loss function. But then we can go one step further, where we can say, well, now we have a concrete model. Like Joshua's group has been working on equilibrium propagation. Equilibrium propagation makes very specific predictions of which variables should matter, and we could perturb them. Like in equilibrium propagation, there's an early and a late period, and it matters if activities go up versus if they go down. Well, we could maybe perturb the system so to make them go up when they would normally go down, and we would have concrete predictions of how activities should change in the brain. And maybe someone else, maybe Blake Richards, has a different algorithm and a different proposal of how the brain does backpropagation, in which case that leads to different testable questions. So basically the field is at the moment opening a lot of like these potential questions, and we will need a lot more physiological experiments. And I should say I'm actively engaged with a group of outstanding physiologists to produce the right experiments, and we're writing grants about that at the moment. Like, keep in mind, like for people who come from computation, that the timescales in biology are slightly different. So if you want to try something different in your deep learning system, sure, you download your Joshua's code, you look at the change, you see if it works. But if you're in biology and you want to do something different, the first thing is there's like lots of modes how things can like spectacular fail. Like your monkey dies, your mouse like never learns the task, like things like that. But even if everything works, it basically means you need to get a team to do that. You need to get the animals. It's like all with approvals and so forth. So basically the relevant timescales, 5 years, where the relevant timescale on the computational side might be a couple of months. But like, yes, we're definitely doing that. Next question. Here we have a question by Isabel. What are those few examples where learning makes you worse? So that prism throwing is a famous case. I throw something, but you give me glasses where it looks like when I throw it, it looks like I throw it into this direction for me. And over time, kind of, I get better at that. And then if I take off the glasses, I've just gotten worse at throwing in the real world, because I will throw in this direction, because I think everything is basically rotated by 30 degrees. And there you have what's called interference, that we have one task that makes you worse at another task. Motor control, the field in which my lab has been active for a long time, has a lot of examples of that. For example, if I make it that if you move here, the robot will push you to the right, and if you move like just 2 centimeters to the left, the robot will push you to the left. You never learn that properly, because being pushed right interferes with being pushed left. But if you then hold the robot like this versus hold it like this, then yes, you can learn that, and they don't interfere. So there's a lot of subtlety about that. Do we have more questions here? Yes. So among the numerous models and hypotheses, how do you decide what you will focus your research on? Are you looking for performance on task, or is it better to explain experimental observations step by step? So this is actually like a meta question, and a question that I thought about a lot. What does it mean for us to do the right research? Like, there are so many questions that we can focus on, and yet we can only work on a small number of projects at a given point of time. How should we choose that? The way I think about that is, in a way, of all the projects that we could run, we should choose those that matter. Now, matter is really complicated. So there are things that we can do that lead to immediate progress. Like, I built a system that avoids cars driving against walls. That's great. It might not be intellectually super rewarding, but it's great because it produces something that is useful. At the same time, there are projects that allow us to think in new ways about things, and they contribute a lot to progressive science. There are also projects that like destroy something that people consider like overly good, and you just basically point out like, yeah, your model can't even do X, or that's kind of a problem. So there are very different projects that can all contribute. So there's not one scale. So the way I choose, the way I mean, like, to me, in a way, it boils down to gut feelings, but you want to be clear. So in my lab, what I always ask people to do is, before they do any experiments, they should write the abstract. So they write the 100-word thing, and then we sit down with the 100-worder, and we're basically, okay, is this 100-worder, if all your research pans out the way you think it is this 100-worder like worthy of you? Is this like the best paper that you could produce? And you often find then that the gut feeling basically tells you, no, I should work on something else. And it might be that it means, no, you should now solve a technical problem. Or maybe it means that the technical problem that you've been thinking of solving is just like too small to be the kind of problem that you really want to spend your life with. So in that sense, there's no easy answer for that. But like, I think you want to be very mindful about that. So what else do we have here? We have here. Have we observed, so this is a question by Mart, have we observed in any experiment that standard spikes are responsible for actions, whereas burst signals are not, and vice versa? So the bridge between stuff happening in single cells and the behavioral level has never, or like has only in extremely limited situations been bridged by neuroscience. So we don't. But what we do have is that there exist certain synapses where they're called facilitating, where basically a burst produces a strong signal, and individual spikes, even if it's the same number of individual spikes, produces very little. And on the other hand, we have depressing synapses, where basically a burst doesn't produce much more than an individual spike. So in that sense, we have observed in experiments that standard spikes may have very different effects than bursts, but we don't know the scope of that. So now, organizers, how are we doing on time? When were we supposed to move on to the next talk?
We have 7 more minutes.
We have 7 more minutes. This is great. So let's see. Okay, cool. So let me try and answer the question by Sana. So Sana asks, the learning methodologies within biological systems are dependent upon the morphology of the systems. In humans, neurological conditions such as Down syndrome affect the capability to learn. How can these physiological differences, morphology and topology, be accounted for meta-learning? Wow, okay, that opens a whole lot of boxes. So let me first point out something that might not be known to everyone here. So there is a field that's called computational psychiatry. Computational psychiatry basically assumes that there's generic computational problems solved by brains or parts of brains, and that parts of this calculation is wrong. So let me give you an example. For example, a theory might be that schizophrenia means that normally if you're a Bayesian, you combine what you know from before, the prior, with what you currently learn, and you combine the two of them into a likelihood. And you can show that it's the right thing. Well, but what if your reliance on the prior was too high? In that case, you can say, I can basically synthesize the world. I believe that the CIA is after me, and I'm so convinced of my prior belief that I am only becoming more and more sure about that. And in fact, I will then start learning from my self-generated data that the CIA is really after me. And people might become very convinced of that. So it's a beautiful field. And people like Quentin Hayes, there's a lot of people who are like in that field at the moment. And in that sense, yes, this is a great point. So the second one is meta-learning. So it is possible that a lot of the diseases of the brain that we're thinking about are like really mistakes of meta-learning. No, like if what's going on in the brain isn't that we just do gradient descent on something. No, gradient descent on something is totally stable. Nothing can ever go wrong with it. But if we have a system where we learn what we optimize and then we optimize the things that we optimize, then there's all kinds of interesting failure modes of that. Now, like, what if the fact that I wrongly learned something changes the way that I learn to learn, and all of a sudden kind of I spin off into some space that isn't necessarily real? So if learning to learn is an important aspect, and I strongly think that it is, then it seems very likely that a good number of diseases we're currently worrying about is affected by learning to learn. Now, like, let's say emotional control. Let's say if we have a learning to learn algorithm, which basically says, if I look bleakly at the world, make it so that I view everything to like more positive ways. And if I look too negatively at the world, let's view everything like, no, if I look too positively about the world, let's make sure that everything gets a little less positive. So if you have algorithms like that, and then the set point of such an algorithm isn't properly tuned, then I may very well basically learn to like move myself into a space where I don't feel well. There's so many aspects to that question. I'm really sorry. I think these are all the things that I can say at the moment. But basically, yes, thinking about those things, like learning might be the answer to a lot of problems for which we currently believe that no answer can exist. We still have 4 questions. What are those? Okay, here. I answered the what about learning making you worse already, I think. Can organisms have multiple learning algorithms, so is backpropagation good enough for learning phenomena? Yes, of course, brains have many ways of learning. No, like it's very clear that the brain doesn't have a centrally represented cost function that optimizes everything. In fact, there's no doubt that kind of cost functions, objective functions change over life. No, like babies can get like really excited about a rattle. If I gave any of you a rattle, chances are like you'd be like, yeah, that's a rattle, and you would very quickly lose interest in that. And like, yet for a baby, like a rattle is like super exciting, and then can go for hours with like, oh look here, it can move. So I believe that the cost functions that are optimized are different across different brain areas. I believe them to be different across developmental stages. I do believe that different brain areas kind of interact meaningfully, so that if one brain area wants another brain area to kind of learn something, then there's a mechanism, maybe like backpropagation, that enables that interaction. But yes, like there is not this, like that there's just one cost function that's simple. No, and it also violates kind of our experience with the world. Like at different points of time, I'm not optimizing Konrad loss function. No, I'm getting myself a sandwich. And now you can say, well, maybe there's like some hierarchical thing, but then what is at the highest level? Like basically like improve Konrad's life isn't very differentiable. And yet sometimes I just know that I want to do make myself a sandwich. And then kind of like I optimize maybe the components of making a sandwich. So clearly it feels like we have different objectives that we switch between. And like, I don't think we can just say that there's like, hey, here's like that simple thing and we just do gradient descent. I want to say, just because I make the point that gradient descent should be in the toolkit of neuroscientists as a meaningful theory of the brain, I'm not reductionist in the sense that I'm like, okay, look, here's like a cost function and then optimize it, and that's it. Now, I do believe that like the brain has this incredible algorithm with which it initializes itself. Now, like Tony Zeta makes a very good point that basically a lot of the behaviors that make us successful are already built into us, and we don't need much learning to get a lot of them.
I have a question, Konrad. Can you make the slides and references available in some form?
Absolutely. Just someone should, if the organizers can send me a link, I'll be happy to send them all the slides.
Great, thanks.
And I'm sorry. There's one more thing that I should say. This was like a very high level overview talk. I'm in no way assuming that that like I'm not missing references. Not like there's no doubt, and I newly wrote it. There's no doubt that there's important references that I'm missing. Okay, cool. Well, thanks everyone for listening in. I look forward to seeing you later at the interactive sessions.
Great. Well, thank you. Thank you, Yusha, for asking your questions, and thank you for Dr. Konrad Kording for such an amazing talk. And so for now, we have our coffee break / social event at Gather Town, and I am just going to send the link right into the chat box. Okay, and so in Online Town, there are labeled rooms for people who are interested in a particular topic, and you can easily walk in and out of the conversation by moving around in the Online Town, and you will only be hearing people who are like virtually close to you. Okay, and so for those of you who want to head over to the Gather Town, go ahead and do so. Otherwise, we will be moving into, we have another session that's called social / coffee break, which means that you can also stay on Crowdcast and we can just play some music and chat in the chat box, whatever you prefer. Right? So thank you guys so much for joining this talk. And then our next speaker will be on at 11 o'clock, which is Eilif Muller. Thank you.